Hello everyone, welcome back to the class. In previous class we learnt about how the membrane proteins, phospholipids are transferred inside the ER membrane with the help of a transport protein called scramblase. And also we learnt why there will be transportation of these phospholipids which are synthesized in the cytosol inside the ER lumen, and the role of smooth endoplasmic reticulum in hepatic cells in removing xenobiotics. And also we came across endomembrane system, which consists of nuclear envelope, endoplasmic reticulum, Golgi complex, lysosome, vacuoles, transport vesicles, etc. And also we learnt the first aspect, that is transport of proteins across the ER membrane. Before revising this part, I would like to give you one easy flow chart. Just a minute. See, to begin with, the endomembrane system consists of a nucleus or nuclear envelope, endoplasmic reticulum, Golgi, lysosome. Right, now we are in, or we learnt about first pathway, that is transport of protein across the ER membrane. ER membrane is interconnected, right? It is having different types or different levels or layers of what membrane in it. The protein which are synthesized in the ribosome or in the cytosol itself, it is roaming inside the cytosol. That protein should pass through these layers of membrane of ER membrane. The first aspect we learnt yesterday was transport of protein across the ER membrane. So it has to move inside this membrane, So first aspect we learnt what? Transport of proteins across the ER membrane, right? Now the second part we are learning is transport of proteins from ER back to cytosol. What we are learning? The transport of proteins from ER back to the cytosol. In yesterday's class we learnt about how the proteins are transported from cytosol to ER membrane. For some reasons, now the proteins has to transport back to the cytosol. The protein has to transport back to cytosol. Why this protein is transported back to cytosol, we'll learn it today. And also, this is the second part. First part was transport of proteins from cytosol to ER membrane, especially across the ER membrane. Next, now we are learning second is transport of proteins from ER membrane back to the cytosol. And also we are going to learn how proteins are inserted into the ER membrane. How proteins are inserted into the ER membrane. This is what membrane? ER membrane. This is ER membrane. How these proteins are inserted to the ER membrane. And also we'll learn in future how the proteins are transported from ER to Golgi. This much aspect we'll learn. From cytosol to ER membrane, from ER back to the cytosol, from insertion of protein into the ER membrane, and also the transport of protein from ER to Golgi. We learnt this first aspect, that is transport of proteins across the ER membrane. That is SRP receptor signal recognition particle, which has two binding site. One end will bind to the signal sequence, other end will bind to the ribosome. It forms a complex of SRP nascent polypeptide plus ribosome. This complex will bind to the alpha subunit of SRP receptor in the ER membrane. The SRP directs both binding of ribosomes to the ER membrane and the insertion of nascent polypeptide into the transmembrane channel. Then there will be interaction of SRP and SRP receptor, directs the SRP to the translocon. Translocon is nothing but a gated translocation channel. N terminal of polypeptide chain enters the ER lumen through this translocon, and next hydrolysis of GTP of SRP and SRP receptor happens, which dissociates the complex or the association. Then the polypeptide chain elongates. So this is the flow chart, how the protein or polypeptide chain are transported, or it is moving across the ER membrane. So next, signal sequence is cleaved by the signal peptidase after its transfer. C63 complex and member of HSP70 family binds BIP are responsible for unidirectional translocation across the ER membrane. BIP has a peptide binding domain and an ATPase domain which binds and stabilizes unfolded proteins. The ATP driven cycle of BIP binding and releases pulls the protein into the lumen. Today we are learning about transport from ER back to cytosol. Why it is happening? See, for example, consider a situation where protein is misfolded, or the protein is not correctly folded. For that, the ER will transport that misfolded protein back to the cytosol. This transport, ulta transport of the protein back to the cytosol is known as reverse translocation, or it is termed as retrotranslocation or dislocation. By this process, the misfolded soluble proteins in the ER lumen are translocated back to the cytosol so that it can be correctly folded, or if it is not possible, it is finally degraded. The first step which going to happen is oligosaccharides are removed, and then deglycosylation is catalyzed by deglycosylation happens, where the deglycosylation means the precursor oligosaccharide we learned, no, precursor oligosaccharide that is removed from the polypeptide chain with the enzyme N-glycanase, and this deglycosylated polypeptide is rapidly ubiquitinated and then fed into proteasome where it is degraded. Let us understand this process with the help of this image. See, first what is happening is this misfolded protein first reacts with the chaperone. If you can see here, it first reacts with chaperone. This is one. Then it reacts with the disulfide isomerase 2. Then it reacts with lectin 3. So first it reacts with what? It reacts with the chaperone. What will be reacting? The misfolded protein first reacts with the chaperone inside the ER lumen. Then it reacts with disulfide isomerase, and the lectin comes and binds to the misfolded protein. This forms a unit now, complete unit. This complete unit is exported back to the cytosol with the help of protein translocator complex. Which is present in this border. Okay. With the help of this protein translocator complex, the protein comes out to the cytosol. Okay, there, while coming out, the misfolded protein or the polypeptide chain is ubiquitinated, means ubiquitin is inserted, ubiquitin is joined with the help of ubiquitin ligase, ubiquitin ligase. Once it is coming outside to the cytosol, the ubiquitin a molecule are added to the polypeptide chain. And also I said deglycosylation. Deglycosylation means removal of oligosaccharide chain or oligosaccharide molecule. So here you can see the oligosaccharide unit, right? That oligosaccharide is removed with the help of N-glycanase. N-glycanase, it is a ATP driven process. After that, ubiquitinated polypeptide chain is sent to proteasome. The ubiquitin which is present in the chain, it acts as a signal for degradation. It acts as a signal for the degradation inside the proteasome. In proteasome, the misfolded protein are then at last degraded. The fate of protein is what decided by this ubiquitin molecule. If ubiquitin comes and binds to the polypeptide chain, it is fixed that the protein will go to degradation cycle. So degradation is happening in the cytosol itself. Why this protein came out? Because it was misfolded. So next, insertion of membrane protein into the ER membrane. Before we learnt this transport from ER back to the cytosol, we learnt one process that the protein has transported from the cytosol to the ER membrane, right? We learnt in the yesterday's class that protein are transported across the ER membrane. So what type of proteins was moving across the ER membrane? Do you remember? It was membrane proteins, soluble proteins, secretory proteins. These type of proteins are transported across the ER membrane. So now these membrane proteins, as the name suggests, it is sent to, or it is designated as membrane proteins as it is present in the membrane itself. So now this membrane protein should be present in this membrane, right? So how this membrane protein are incorporated in this membrane, we are going to learn in the next slide. Soluble proteins are also embedded inside the membrane. Secretory protein has different fate now. Membrane proteins are what decided that membrane protein should present in the membrane, should be inserted into the ER membrane itself. That aspect we are learning now. Insertion of membrane protein into the ER membrane. Integral proteins of ER, Golgi, lysosomal membranes and in the plasma membrane are synthesized on the RER. These proteins are initially inserted into the ER membrane. From ER membrane, they proceed to their final destination along the same pathway as that of secretory protein. From ER, it enters to Golgi and then it enters to the plasma membrane or lysosome. This is membrane proteins which are to be destined to the plasma membrane. ER membrane, whichever proteins which are needed for ER membrane, it will settle down in ER only. So which are the types of membrane proteins we learn now. transmembrane integral proteins. Before getting into this topic, I want to clear one thing. Integral protein and intrinsic protein and extrinsic protein. If you see, the plasma membrane is having hydrophilic head and hydrophobic tail. Hydrophilic head and hydrophobic tail. This is hydrophobic tail made up of fatty acids. The intrinsic protein are embedded inside the plasma membrane or the membrane. Extrinsic protein are present at the border. Extrinsic protein are present at the border. Okay, this integral protein should be inserted into the membrane only. So it is inserted. Okay, so this is the difference between intrinsic protein and extrinsic protein. So we are learning about integral protein. See, it is embedded. Everything is embedded here. So they are termed as integral proteins. These are proteins that are embedded in the membrane by hydrophobic region that spans the lipid bilayer. These membrane proteins have 4 topological classes: type 1, type 2, type 3 and type 4. So this is single transmembrane protein with C terminus, N terminus. We'll learn it in the next further slides. The N-terminal signal sequence on the nascent type 1 protein initiates translocation of the protein. How these proteins get embedded? So there is a N-terminal signal sequence which allows or which initiates the translocation of the protein. N-terminus of the growing polypeptide enters the lumen of the ER. Which end will enters the lumen of ER? The N-terminus of growing polypeptide enters. Next, the signal sequence is cleaved. Then the sequence of non-polar amino acid, that is stops transfer signal, the pink one here, stop transfer sequence will be cleaved, so that it will not allow the entrance of that protein further inside the lumen. This internal sequence moves laterally and become anchored in the lipid bilayer of the membrane. Then translation continues at the ribosome. As C-terminus of the protein chain is synthesized, it loops out on the cytosolic side of the membrane. See here, the ribosome is still synthesizing the protein, right? The polypeptide chain. The N-terminal growing polypeptide will enter the lumen, and there is a stop transfer signal where it allows the stoppage of transfer of the growing polypeptide. Still the ribosome synthesizes the polypeptide until the C-terminus is synthesized. As C-terminus of the protein chain has been synthesized, it loops out on the cytosolic side. Making that protein to embed inside the membrane.